Okay, so we left off last time about talking, just to review very quickly, right? We talked about this very unusual situation where the genetic testing would suggest that the mother wasn't the mother, but the father was the father. And we described this story to you a little bit. And really, part of the reason for illustrating it to you is that there are a lot of exceptional things that happen in biology. So obviously we teach you how things normally happen. But in fact, the way we know very often how things normally happen is by knowing what happens when they go wrong. Recognizing those unusual situations is really how we understand the rules. And so this is a particularly good example, and I told you that there was another woman who had the same sort of issue with genotyping her and her sons, and that in both of these cases, these women turned out to be human chimeras, right? So there was really nothing wrong with the DNA testing, but there was something interesting going on in the biology that was unexpected. In this case, they were the products of fusion of fraternal twins. So normally the twins obviously stay separated, but occasionally they will fuse. Sometimes they fuse very late in development, and then you can get serious issues like this, where it's actually a conjoined twin, but what's left of the second one is just the arms and legs here. And in this case, this girl was successfully separated from those extra appendages. And in the case of these people, they were chimeric, but most of the chimeras, they looked completely normal because the fusion had occurred very early, and those cells of their twins had been incorporated. In Karen Keegan, they were incorporated in a fairly major way. It was easy to see that many of her tissues were mosaic, whereas in the case of Lydia Fairchild, it really was almost only her germline that had come from her twin, and that's why it was actually very hard to detect. Okay, so now I'm going to do an iClicker question. So go ahead, and also get out a scratch piece of paper. To write down on, or any piece of paper, you're not trying to get in or anything, to write some numbers down on, okay? And let me describe what we're going to do. Okay, so you're going to see these numbers, and this is kind of an experiment. Can we turn the lights down a little bit? Is there any way? No? Okay, good. So what we're going to do is I'm going to show you on several slides in a row, I'm going to show you, there will actually be 2 of these circles on each slide with numbers in them, a 1 or 2 digit number, okay? And now, first of all, there's no right or wrong answer, so one thing I want to ask is that you not ask the person next to you what they see, okay? It's only what you see, all right? And this is kind of an experiment. I don't know if this is really going to work. It depends on whether the color balance of the projector is really right, and I kind of don't know. I maybe doubt it, but we'll see what happens, okay? So there'll be 2 circles at a time. Write down the numbers you see in those circles, and I'll give you about 6 seconds per slide, okay? Okay, so here's the first set. Okay, the next one. Okay, next one now. Okay, one more. Okay, this will be the last one. Okay. All right, so now you're going to, just in a minute, I'm not going to turn on the iClicker quite yet, but what I'm going to ask you to do is figure out what you saw, okay? And it's just you, don't ask anyone next to you, all right? Write down whether there's A, B, C, D right now, because I'm going to take it off for a second in order to turn on the iClicker, okay? Okay, so, oops, sorry, got to plug it in. Okay, you can start voting now. That's why I said to write them down. Okay, sorry. Here. Okay, a few more seconds. Okay. Okay, now let's see if this works. Okay. So it's interesting. So it didn't quite give what we were expecting. That's okay. So, but it gives the basic idea that you can see that most of you can see the numbers, okay? But if you look percentage-wise, okay, the males, right, are more likely not to be able to see the numbers than females, right? I know it says that the number looks the same, but of course there were many more women voting than men, okay? So the percentage for men is slightly higher than women, okay? And this is a standard colorblind test, okay? And it's mostly testing whether you're red-green colorblind. So the question now is then, let me go ahead and shut this down. Okay. Okay, so why is this, okay? So in humans, colorblindness is more common in men than women, and we saw that in that. If you looked at it, I don't know, you could do a test, it was really statistically significant, but there's a slightly greater percentage. Usually the percentage is really quite different. So it's usually about 7 to 8% of men are colorblind, red-green colorblind, and only less than 1% of women, okay? And so it's actually genetic, okay? So it's interesting to have something that's genetic, but that's biased towards one sex, okay? And remember that when Mendel did his experiments with pea plants, right, he didn't observe any differences between inheritance from males and females. That's a little complicated. The plants are both male and female, but it didn't matter which pollen and which eggs he used, basically, right, which stamens and pistils he was using when he did his crosses, right? So when we showed these kinds of crosses, it didn't really matter which plant, which flower provided the pollen, okay? But what we know is that, in fact, there are differences, genetic differences between males and females in a number of animals, not in everything, and we'll go over that, okay? So, for example, in Drosophila, right, in fruit flies, there are 4 chromosomes, okay? There's 3 autosomes, and 2 of them are pretty large, one is very small, and then there's also a sex chromosome. And that chromosome, what happens is in females, there are 2 X chromosomes, but in males, there's 1 X and 1 Y, and they look quite different from each other, and the X is larger than the Y, okay? And in fact, the discovery of these chromosomes and the fact that they weren't the same between males and females led to basically important evidence that genes were really on these chromosomes, and that was by Thomas Hunt Morgan. And he discovered a mutant in flies called white, okay? And he showed that it had an unusual mode of inheritance. So the phenotype's pretty striking. So this is a normal eye color, and this is a mutant, homozygous mutant for white, okay? And I say homozygous, but you'll see what the complication of that is in a minute, okay? And what Morgan did is he mated flies with white eyes, okay, with female flies with red eyes. So he took males with white eyes and he mated them for females with red eyes. And as you might expect in the F1 generation, they all had red eyes, right? It's consistent with it being a recessive mutant. In the F2 generation, he got the expected 3-to-1 phenotypic ratio, okay? But only the males had the white eyes, okay, and none of the females, okay? And so Morgan determined that the white-eyed mutant allele must be located on the X chromosome, and that this pattern of inheritance fit the pattern of inheritance of the X versus the Y chromosome, okay? So we can illustrate that here, okay? So if we have white is a recessive, right, and white plus is the normal allele. So again, if he does a cross between white plus and white minus, he gets white-eyed male and a red-eyed female. All the progeny have red eyes. Okay, so that seemed to be okay. Oops, wait, sorry, to make this, yeah, that's right. Okay, and then, but when he mated those progeny together, again, you had the 3/4 red eyes, 1/4 white eyes, but the white eyes only appeared in males. In fact, it was half the males had red eyes, half the males had white eyes. Okay, so how is this? And so it's what you expect in one sense for the phenotypic ratio, but why is it sex biased? Okay, and again, this just illustrates that cross. The first set of offspring of a red-eyed female to a white-eyed male, everybody has red eyes in the next generation, but in that F2 generation, you get this pattern where it's 3 and 1, but these are all female, then this is male, and this is male. So the white eyes only appears in males. Okay, so probably again, if you're taking the lab at the same time, you know quite a bit already about how you get segregation on the X. So again, what's happening, here's some other crosses, right? So if you took a white-eyed female and a red-eyed male, okay, then you would get half red, which were all female, and half white, which were all males. If you took a white-eyed male and a red-eyed female, again, he got all red eyes. But right here, you see a problem, is that you see that it depends, the outcome, even in that generation, depends on whether it's the male that has the red eyes or the female that has the red eyes in this cross, okay? And so this was kind of inconsistent initially with Mendel's rules, where, Mendel didn't say that there was any reason to worry about which was the male and female in his crosses, okay? And so we understand this now, and we know that X is on that white, the gene, is on the X chromosome. And females have 2 copies of the X chromosome, whereas males only have 1 copy of the X chromosome. And the Y chromosome actually has very few genes on it, and it doesn't have the white gene. It only has a few genes that are required for male fertility. Okay, so that means that any genes that are on the X, the male is dependent on that 1 copy of the X for whatever it can get. All right. So in this case, if you had a red-eyed male, it'd be white plus over Y. Okay, and a white-eyed female would be homozygous mutant for white on the X chromosome. So you can see now, if you cross these, what's going to happen is you're going to get all the progeny with 2 X chromosomes. So one of the X's comes from here, one of the X's comes from the female. Those are all female, and they all have red eyes because one of their copies of the X was from the male, and the only copy that the male has is a white plus. Okay, whereas if you look at the males that result from this cross, so one chromosome coming as a Y that's coming from the male, that means the other X chromosome has to come from mom, and she only has white minus chromosomes. So these would be all white-eyed males. Okay? And so that gives you this unusual pattern of inheritance because you have this linkage on the X chromosome. Okay? And so again, you can write this out for yourself in the same sort of way as you do a Punnett square, and you can see this. So this is for the other cross now, where we have the heterozygous, and then we're looking at the F2 generation. And again, we got 3/4 red, 1/4 white, but you can see all the white individuals are males, okay, because they have a Y chromosome, and then the only white minus chromosome, the only X chromosome they're inheriting is white mutant. Okay? So that gives you that sort of pattern that you get. So real quickly now, why don't you try this? So try these crosses and see what you expect for the outcome. So what you want to know is both the phenotype and whether they're male or female from these 2 different crosses. Okay, I'll give you another maybe half minute. Right, make sure. All right, so this is hopefully what you got. So if you do this now, where it's female white plus white minus to a male that's white plus Y, okay, what you should find is that all the females have red eyes, okay, and they're either white plus or white minus white plus, okay, because they're getting this X chromosome from dad, and then they're getting one or the other of these, but it means that they're going to be red-eyed because they've got at least 1 white plus chromosome. Then in the males, you're going to see that they get the Y from dad, and they're going to either get a white plus or a white minus chromosome from mom in equal ratios, so the male's half a red, half a white. And then in this cross, okay, now again, the female is heterozygous, but now the male is mutant for white because he only has the 1 X chromosome, it's white minus. So now you're going to get these possibilities here. So you can get females that are red-eyed, that are heterozygous, females that are white-eyed, that are homozygous white minus, okay? And then in the males, again, you get the same thing. You get males that are white-eyed and males that are red-eyed. So again, they get Y from dad, and they either get a plus or a minus white from mom. Okay? So that's the kind of ratios you get when you're on the sex chromosome. Okay? So Morgan observed that the patterns of inheritance could be explained by the hypothesis that the gene was located on the X, because this is what people had observed for the segregation of the X and Y chromosome, that the Y was always coming from dad. Animals that were XY were males, animals that were XX were females. Okay? And so the mode of inheritance of white plus and white minus could be predicted by the behavior of the X chromosome during meiosis and subsequent fertilization. Okay? So that's the sort of simple variation on Mendelian genetics. So again, it's an example where first you do the cross, you have what looks like some sort of violation of Mendelian genetics, but when you understand a little bit deeper about the biology, in this case that the genes are on the chromosomes also control sex determination, and in males there's only 1 copy of that particular chromosome, okay, that's actually carrying the white gene and not 2 copies, then you understand that pattern of inheritance. So it turns out that in humans you also have a sex chromosome. You have an XY system as well. So we talked about in humans how you have 22 autosomes, and then the 23rd in this nomenclature in humans, the sex chromosome is the last one. In flies, it's actually just by the way it's named, it's the first one. But anyway, in humans, it's 23rd chromosome is the sex chromosome. So this is a female, right? And in that right there, she's XX, and a male is XY. And you can see the Y is a very small chromosome, okay? And the other important thing is that these chromosomes don't undergo recombination in the males. When there's an X and a Y, you don't get recombination between those, okay? So the X stays, in essence, separated, okay? And you can inherit the X or the Y, but you don't have any alleles to recombine with on the Y. All right, so this actually is the reason for color blindness being predominant in men in humans, okay? And it's because the mutations that give rise to the most common form of color blindness are on the X chromosome, okay? So you get this kind of pattern of inheritance. So in this case, the green stands for a wild-type allele, a normal copy, and the blue is the mutant copy, the one that's not able to properly see in color. And so what you see is you have an unaffected mother and affected father, right? So he's only got one copy of that. And that's why he's colorblind, right? Because he only has one copy, and the one copy he has is defective, all right? So in these children, then what happens is that the sons are unaffected because they're getting their X chromosome from mom, so they're getting their Y from dad. The daughters are all carriers. They're all heterozygous, okay? But they're not colorblind, okay? And why aren't they colorblind? Because they have a normal allele as well, okay? And the colorblindness acts as a recessive mutation, okay? But on the other hand, if you have a mother that's a carrier, right? So if one of these daughters grows up and their spouse is unaffected, okay? Now you see what happens here is that half of the male children will end up being colorblind. And the reason is because, again, they're getting their Y from dad, but they're going to get one or the other of the X's from their mom. And if they get that X that has the mutant copy, right, that's the only copy of that gene they have, and they're now going to show the mutant phenotype. Even though it's a recessive mutation, there's only one copy of that chromosome, so you're going to see that, okay? And the reason this works out in humans is that you have 3 photoopsins, so you have these special molecules in your photoreceptors in your eye that are designed to perceive wavelengths of light. And you have a blue, a red, and a green receptor, basically. And the blue one is on the autosome, okay? And it's actually pretty rare for there ever to be a mutation in that gene, so most people have no problem with that one. But it's the red and green one that causes the problem. And so on some X chromosomes, there's a deletion of either the green one or the red one. And later on, in the rest of this week, we'll get around to explaining to you why this is so common in humans, too, why the deletion occurs so frequently. But one or the other will get deleted, okay, every now and then on an X chromosome. And so then these are women. If this is in a woman, then she's simply a carrier, right, because she'll have a normal copy of the other X. But if a man gets this particular mutant chromosome, then he either can't see red, he can't distinguish red from green, because there's only one photoreceptor there in that red-green wavelength area. And so what happens is that if you have that number, that's just shades of reds and greens, then you have a hard time seeing it if you're colorblind, okay? And the reason that, again, it's more prevalent in men is because men have just one copy of the X, and so they're more likely then to be affected by this, okay? So we say that genes that are located on either sex chromosome are called sex-linked genes, okay? And genes on the Y are called Y-linked, and genes on the X are called X-linked, okay? And again, the Y doesn't have very many genes on it, okay? So, and then as we just showed in Drosophila, the X-linked genes follow specific patterns of inheritance, and you can then work them out, right? We've just gone through the different examples that you can have in terms of crosses, okay? And so for a recessive X-linked trait to be expressed, okay, just as if it were on an autosome, it behaves that way in a female, that you need to have 2 copies of the recessive mutant allele. But in a male, you would only have to have 1 copy of that mutant allele, okay, because the Y doesn't have copies of those genes, okay? And again, this is why X-linked disorders are much more common in men than in women. And there's a number of diseases or different phenotypes that show up in men more frequently than women because of this in humans. And one we've already talked about is colorblindness, where most of the mutations that cause colorblindness are X-linked. And then also a particular form of muscular dystrophy is X-linked, and hemophilia is also X-linked. So all of these are more likely to show up in men. And women that are carriers, right, are going to have half of their male children will generally be affected with the phenotype then. Okay. And as we said then, okay, and it was this discovery of the segregation pattern, first in flies and also in other animals later on, that was part of understanding this chromosomal basis of both the determination of sex, but we'll talk about, that it's not the same in every animal, okay, and that genes were really on these chromosomes. So in humans and other mammals, there are 2 varieties of sex chromosome, a larger X and a smaller Y. The ends of the Y chromosome do have regions that are homologous with the corresponding regions of the X, but mostly they're devoid of any genes. But there is a critical gene on the Y, so there's a couple of genes that are required for male fertility, but there's a key gene called SRY, which is the sex-determining region of the Y, okay, and this is just for mammals. And it's actually this individual gene on the Y that codes for a protein that directs the development of an anatomical male, okay. So if you have that little, it's only that little piece of the Y you really need, and that part of the Y will actually direct the embryo at a very early stage to become male. It'll actually develop testes and begin to form male sex hormones and end up as a male, okay. And here's just an EM image of the X and Y. You can see the X is much, much larger than the Y chromosome, okay. So again, females are XX and males are YY. And what we did with that Punnett square, right, is to show you that each ovum that a female is making contains an X chromosome, okay, because that female has just 2 X's, right. But the sperm can carry either an X or a Y, okay, because the male doesn't have 2 X's, he has an X and a Y, okay. But it turns out that you've learned about flies and you've learned about humans, right, and they have this XY system of sex determination. Do you know any other mechanisms for sex determination? We actually talked about one already. Temperature is one. What's the one we talked about? Does anyone remember that? What's that? Yeah. So the one we talked about had to do with whether the egg was fertilized or not, right? So that was in ants and bees and wasps and what we call Hymenoptera. You get a haploid-diploid type of sex determination. But it turns out that there's many ways, actually, to go about working out sex determination. It seems like a very odd thing because it seems like having a sort of a reasonable ratio of males and females is really important to the survival of the species. And you often think that, yeah, is there a question? Yeah. I'm sorry, say it a little louder. Okay, so it depends on the type of species that you're in, and it gets very complicated. But as an embryo, if you make male hormone, it masculinizes the entire embryo, even if it's XX. Okay, so it's really the sex-determining region of the Y. It gets incredibly complicated, and a lot of unusual things can happen both genetically, and then there's a whole different aspect of the the sexualization of the brain. A lot of the things in the brain work very differently too, and so we don't even know how all of that works yet. Okay, but this is if we think purely anatomically, whether you're male or female. Okay, it's controlled this way. It gets complicated because like left-right asymmetry, there's a certain component of it that's genetic, right? But there are other components of it that are not genetic, and so this sex, of course, is a very complex thing. We're talking here just about from an anatomical viewpoint. Okay. Okay, so there are lots of different ways, though, that you can have systems for sex determination. Yeah. Yeah. Yeah, we won't go over that yet. We'll go over that later. There are other possibilities, right? And we'll talk about that not today, but we will talk about what can happen, right? And like I said, it gets